Back again with the mini lecture on tonicity. I know you just did a reading on this, but um, this concept is so confusing for some students and even some of the teachers that I work with, professors that I interact with. I see them misconstruing this all the time. So I just want to make sure you really understand tonicity, osmosis, diffusion, right? How molecules, water, solute, solvent, what are we talking about? So I made this little lecture on tonicity, which I hope is helpful and may clear up any misconceptions that you have after the reading. Okay, so tonicity describes how an extracellular solution can change a cell's volume by affecting osmosis. So we talked about diffusion, we're talking about diffusion of molecules. When we talk about osmosis, we're talking about diffusion of water. When we're talking about tonicity, we're talking about how the diffusion of water or osmosis affects the solution, the solute plus the solvent. Let's get into it. A solution's tonicity often directly correlates with the solution's osmolarity. Osmolarity is the solution's total solute concentration. So the total concentration of solute in a solution is called its osmolarity. A solution with low osmolarity has a greater number of water molecules relative to the number of solute particles. We see that here on this side in this figure from the last mini lecture. So here there's more water than there is solute. So this solution on this side of the membrane has low osmolarity. On the other hand, a solution with high osmolarity has fewer water molecules with respect to solute particles. So we can see that on the right side of this diagram. There's a higher concentration of solute particles than there is free water. So the osmolarity is high here. In a situation where a membrane is permeable only to water, which is what we saw in this, that's the semi of this semi-permeable membrane, it only allows water to pass through. So in this kind of situation, um, the solute separates two different osmolarities. We have low osmolarity on this side and high osmolarity on this side. So water is going to move following the rules of osmosis from the membrane side with the lower osmolarity down the concentration gradient to the side with higher osmolarity. So from more water to less water. This effect makes sense if you remember that the solute cannot move across the membrane in this particular instance, and thus the only component in the system that can move is the water. The water moves along its own concentration gradient independent of the molecules, independent of the solute, okay? An important distinction that concerns living systems is that osmolarity measures the number of particles in a solution. Therefore, a solution that is cloudy with cells may have a lower osmolarity than a solution that is clear. Okay? If the second solution contains more dissolved molecules, then there are cells. There are three types of tonicity. A solution can either be hypotonic, hypertonic, or isotonic. What does this mean? In a hypotonic solution, the extracellular fluid has lower osmolarity than the fluid inside the cell. So here we have our cell, which is outlined in yellow with a yellow plasma membrane. We have red solute molecules and blue water molecules. We see that there is more water outside of the cell, so in the extracellular space, than there is solute. So this means that this fluid has a lower osmolarity than inside the cell. Okay, so if this happens, and we know that we're talking about water, and we know that it wants to be a dynamic equilibrium, we know that the water is going to move into the cell. It's going to rush in there even as the solute moves out. In this situation, water will follow its concentration gradient, moving from high concentration area outside the cell to low concentration area inside of the cell. And what will happen to the cell? It's going to explode. It's just like putting too much water in a water balloon. Now let's look at a hypertonic solution. In a hypertonic solution, here we have another representation of a cell. Remember, the prefix hyper means more. So this is when we're talking about hypo and hyper, we're referring to what's happening in the extracellular fluid, right? The solution. That's why we always say hypotonic solution, hypertonic solution. The solution is what is outside of the cell. Okay? So in a hypertonic solution, the extracellular fluid has a high osmolality, meaning that there's less water outside of the cell than inside of the cell. Because the cell has a relatively higher water concentration, and it wants to be a dynamic equilibrium, it's going to follow the rules of osmosis and diffuse out, right, from the intracellular to the extracellular. This is going to cause the cell to shrivel, which we call crenellation. In an isotonic solution, iso meaning equal, if you remember that from physics, maybe chemistry, the extracellular fluid we see has about the same osmolality as the inside the cell. If the cell's osmolality matches that of the extracellular fluid, there's no net movement of water into or out of the cell. And although water will still move in and out, it's doing so at equilibrium, okay? So blood cells, plant cells in hypertonic, isotonic, and hypotonic solutions take on these characteristics or appearances. You can see this if you haven't watered your plant in a while, the leaves start to droop, and that's because what happened? There's more water in the outside in the inside environment than in the outside, right? So there's more water in the cells of the plant than outside of the cells of the plant, which causes the leaves to be heavy. If you put too much liquid, let's say like if you have a salad and you soak the salad with salad dressing, then you're going to have, what you're going to see is that the leaves start to look broken or discolored. That is because all that solute rushed, all that water solution, the salad dressing, rushed into the cell and caused the cells to explode.